Hello and welcome to a very special series of Dive into Deeptech Moss, brought to you by Republic of Work and powered by HelixWorks Technologies. I'm your host, Eitan Yaakobi. For those unfamiliar with MOS, it stands for Molecular Storage System. In other words, using DNA. Yes, you heard it right, DNA as a data storage medium. We're excited to explore the incredible possibilities of storing data on DNA for various purposes beyond digital archival. Throughout this series, we will be talking to experts and innovators in the field to get their insights and perspectives on the potential of using DNA data storage. We will introduce the basics of DNA data storage, discuss various technologies used to store data on DNA, address the challenges in using DNA as a form of data storage, and explore its potential applications in different industries. So join us as we dive into the exciting world of DNA data storage and explore the potential of this revolutionary technology. I'm so excited to bring another episode of the series. Today, we're joined by Professor Zohar Yakhini and Professor Eitan Yaakobi. They both are leading breakthrough research in DNA data storage at Tech9, Israel Institute of Technology. Professor Zohar is currently a faculty member in computer science at the Interdisciplinary Center in Herzliya and visiting faculty member at the Computer Science Department at Tech9. He leads an active bioinformatics research group with students in both the institutes. Prior to joining academia, he has been a master scientist at Agilent Laboratories until April 2016. Professor Eitan is an associate professor at the Computer Science Department at the Tech9. He leads the DNA Data Storage Lab, and he's currently the head of Tech9 Excellence Program. Welcome to our show, Eitan and Zohar. Delighted to have you join us. There is loads and loads for us to uncover and chat about today, but I'd like to start with a bit of an introduction and what got you hooked to DNA data storage. If you'd like to go first, Zohar.
Okay, thank you, Eshna. It's a pleasure to be here. Thank you for hosting us in this podcast. Yes, so I did, my background is mostly in math and computer science, my educational background. So I did my bachelor's in math and computer science and then continued to do my PhD at Stanford in California in very theoretical aspects of probability theory. I'm always saying that I did not know how to spell DNA when I finished my PhD, which is almost true. But right after my PhD, I started to do research in computational biology, and that's where I stayed. This is my research interest currently also.
And Eitan, you also have a background in both mathematics and computer science. How did you transition into DNA data storage, and what interests you the most about this field?
So first of all, thank you, Ashna, for the invitation and hosting us today. I guess we are both very excited to take part of this podcast, and we're very happy to share what we've done so far in DNA storage and also maybe chatting about the future. A little bit about my background. I did my undergraduate degree at the Technion in computer science and math, and then I continued for a master degree also at the Technion under the supervision of Professor Tuzi Etzion. Then I moved to UC San Diego to do my PhD with Professor Paul Siegel, Jack Wolf, and Alex Vardy. And that was under the Department of Electrical Engineering, but pretty much in the same area of information theory and coding theory. During my postdoc at Caltech with Professor Shuki Vrook, I was also around similar topics of, for me, like everything was around storage and memories. So in fact, during my PhD, I was mostly interested in coding for flash memories, solid state drives, and so on. It was relatively new at the time, so it was very exciting. And I also took a big part of it during my postdoc. And shortly after I joined the Technion, I think I had a short crisis in my career in terms of, okay, now I've got a position, but I need something new, exciting to work on, because it's nice that you have something which it's not only theory, but also has application. So I heard about DNA storage already during my postdoc, but I wasn't understanding enough what it means and how it's actually being done. And maybe around 2015, 16, one of my master students at the time, she expressed interest in DNA storage. And I thought, wow, it sounds cool. Like, I mean, let's try to see what could be done about it. So then we started researching. I remember at the time I was talking a lot with Professor Olgita Milankovic from UIUC, and she helped me also a lot to get into this field. And I think very shortly after that, I fell in love in this area. So as of today.
And just in terms of the areas of interest to your each group, you're both leading to different groups in DNA data storage at the Tech9. Would you be able to give us a quick overview of the Yakhini group, Zohar?
Yes. So as you said before, my research group is basically consists of people, students working in two institutions. One is the Technion in Haifa, and the other one is Reichman University, or the Interdisciplinary Center in Herzliya. And I have people from both institutes working in what I consider to be a single research group, and we're working together. So as I indicated, I started working in computational biology in general, not in storage in DNA. That only came a little later, of course, because this did not exist when I started when I finished my PhD in 1997, this idea, I don't think really existed, or at least it was in very preliminary stages. But I started doing research in the industry, really. I worked at the time, it started as a group inside Hewlett-Packard, and then spun off to be Agilent Technologies, which took over the non-computing business of Hewlett-Packard, including life science measurement. And this is where most of my research focused for many years. My current research interest, or the research activity of my group, I would say fall into four main categories. One is synthetic biology in general, and the use of synthetic DNA for storage in particular. So this is where our conversation today resides mostly. Another is maybe another aspect of synthetic biology, but I think it has implications beyond synthetic biology as well, which is genome editing. So we're looking into different computational aspects of how to measure and how to improve genome editing. CRISPR mostly, but not necessarily only CRISPR. A third very active area, which is probably the one that historically is the oldest research area for my group, is I would say, general computational aspects of molecular biology. So this can go from statistical approaches to measuring differences in DNA or RNA, or understanding transcription factor binding through computational tools, and developing methods and analyzing data in these areas, with a particular interest in cancer research. So we have a lot of interest in cancer research, and from that interest in cancer research, one of the areas that has grown as work in my group is the analysis of medical images, so mostly pathology images related to cancer. So we're looking at pathology images and are trying to understand what it is that we can infer from these images. So these were three areas. And then the fourth area would be the development of general statistical and machine learning methods, either with applications in molecular biology or with applications in other fields. So I think that's mostly where my research interests and the work in my group is happening. So it's mostly applications of computational methods, machine learning, statistics, data science, mostly, but not only molecular biology.
That's quite diverse, and I think that's the future of technologies as well, working with cutting edge tools and technologies at converging sectors. And what about yourself, Eitan? You are leading the DNA Data Storage Lab and also the Tech9 Excellence Program. Can you briefly outline what the program is about, as well as what's happening at the DNA Data Storage Lab?
Good. So in terms of my group at the computer science department at the Technion, So maybe I did not excel as Zohar did with understanding biology and many other topics. So my knowledge of DNA is pretty much in the level of A, C, G, and T. I don't know what the difference between DNA and RNA. So I'm focused more on the coding and the information theory side, and this is where my background is from, starting my PhD. So basically, in my group, what we are doing is error correcting code, other coding techniques, information theory for several applications. So the first and the major application is memories and storage, and that could be relevant both for DNA storage, but not only. So I did a lot of work on the flash memories, solid state drive, hard disk drive, race-track memories, memory stores, and basically almost every storage technologies. Also, when we think about storing data in the cloud, what's called like erasure coding for storage applications. So from the system point and level. So also coding for privacy and security. So pretty much what we're trying to do is to identify where coding could be helpful in many applications, not only storage, and to see what we can contribute and do. Many of the work that I'm doing are purely theoretical, and I'm very excited about them. And it's nice, even though you know sometimes it sounds like it's not as useful, but I tell my students it's useful because they can write a paper, so it's already one good application. But we know that things that even when we get to practice, things should start from the theory, and that's what we're trying to develop. Even today with DNA storage, many of the work that we are doing is completely theoretical, and we're happy about it. And many things which we're trying to do is to really, in my lab, is to really to do wet experiments and to see that the impact is not only theoretical. Because one thing which I learned during the years is that in many topics you have two groups. The ones who actually do they do everything from the device, the application, they build the systems, and then you have the ones who are doing the theory. Now the question is who is talking to who and who should come first. And sometimes when the theoreticians can be more of like on the snobby side, and they think that the ones who actually build the system should come and talk to them. And I have to admit that I used to think this way in the past. And one thing which I learned during the years that it does not work this way. And if you really want to make a true impact, we need to reach out and to really get into the system. And if you really want to design something, you have to understand the fundamental limits and how things work and exactly what the constraints are, and to do the full system of solutions. And that should also be as part of collaboration and finding the right collaborators.
That's actually quite interesting, because that kind of brings me to the next part is that both the Yakhini group and DNA Data Storage Lab might be collaborating on different aspects of DNA data storage, and we'll kind of moving forward, we'll try to keep the focus on DNA data storage as much as synthetic DNA can be employed for a lot of things. We'd be touching that as well. But how do you see the collaboration working between your research groups in Tech9, and what role do you think collaboration plays in bringing such technologies to market as well?
So maybe I'll start where Eitan stopped in his previous discussion, description. There's a saying that in theory, practice should follow theory. In practice, it doesn't. So I think in our collaboration, practice does follow theory, but to a certain extent. And I think a good example for that would be our work, the work from my group, which was led by Leon Hanavi, now Dr. Leon Hanavi. He did this during his PhD, where we used what we call composite DNA letters to improve certain aspects of the density of the synthesis. We can talk about it a little bit later, maybe. But I think the theoretical ideas, they were nice. We could prove things on paper. We could calculate things on paper. But then to make a real impact, as Eitan was saying, we had to go and actually do that large scale. And part of the reason is that when you do it large scale, things that happen once in a million, because you have 1.6 million oligos, and each one of them is linked to 150 cells, these things that happen once in a million, they happen. And they happen a lot of times. And you have to deal with these questions. And you have to have had a theory that allows you to deal with these questions because you anticipate them. And this is exactly where coding theory, for what Eitan is doing, is coming in, and to do it in the right approach. So I think I'm also going to use this as an example for how the collaboration works between us, because we did something very naive in that work in terms of coding theory. And it's only after the fact that we came and talked to Eitan, and we realized that we could do the coding, the error correcting coding, in a much more principled and an approach that would be a better fit for that particular application. And because we are not experts in coding theory, we know the basic concepts of coding theory, but not as advanced as what Eitan is doing in his group. So putting these things together, we can now apply the correct error correcting coding approaches when we face the next phase of trying to make synthesis or storage even more efficient. And I think this is the big advantage of the collaboration between our two groups. And we have, since we started with the first project that goes along these lines of doing reconstruction algorithms that are motivated by a certain synthesis method, or doing coding schemes that are motivated by other synthesis methods, etc., we're finding more and more questions that actually can benefit from both statistical, computational, molecular biology ideas, as well as from deeper understanding of coding theory, which is what Eitan brings to the table.
So what are some of the projects, if you'd like to highlight, where both of the groups have come together, besides the one that you mentioned? Are there any more projects in the pipeline?
Yeah, so maybe I'll say my side of the story for this love story. So we started working together about 5 years ago, and really the motivation came. I had a master's student at the time. Now he's doing a PhD, and he's still working both with me and Zohar, almost Tabari. And when he started doing his master, he did not want to do only theory. I was more theoretical at the time, and he wanted to do DNA storage, but something which is more applied. I also wanted to do it, but also knew that my knowledge is quite limited. In 2017, we had a conference at the Technion, and Zohar was one of the speakers. So I think that was my first interaction with Zohar, and I knew at the time he was going to be my collaborator. So it was only a matter of time. So we reached out to Zohar, and together with Omer, and we did some work also with now Dr. Leon Anavi. And the first project was, and that's one thing which I wanted to understand first, is to understand how the errors behave in DNA. So say that we run some experiment, and we know what we want to synthesize, we know what we read, but if you want to design a good codes to correct errors, you have to understand what the characteristics and how the errors behave. Now you can do it every time whenever you do it. So you run some experiment and then you try to analyze how the errors behave. But what we thought is to build a tool that will help not only us, but all the community, in the sense that if now you want to come and run some experiment and you want to understand how the errors behave, we don't want you to work so hard. So we will give you all the tools, and that's what we have. We have a tool which is called SOLQC. And the idea is to have like you come to a doctor, and the doctor will tell you where you're sick or what's wrong. So we tell you exactly how the errors behave, and we try to give as much statistics as possible. And that was the first work that we did. And then we did some more work about the reconstruction algorithms. And with Zohar as a student, now he's a PhD student, Roy Shafir. And that was also a joint work together with Omer Tabari, to build reconstruction algorithms for a specific DNA synthesis method, which is, we call it stutter noise. So that's something that started with a group of George Church. And the type of the dominant errors have a specific behavior in which every base could be elongate. So instead of synthesizing only one A, you can synthesize more than one, or you could also have what's called the deletion. So then to design specific algorithms for this kind of noise, that was another work that we did. So that under the reconstruction algorithms. For me personally, one thing which I really like a lot is the work that Zohar did about the DNA composites. And it was very nice. I remember the time when I first heard about it, I did not understand how it works. It took me some time. But once I did, I understood what the high potential that we can have using a composite DNA synthesis. And currently, a big part of what my group is doing, some work is done also together with Zohar, is to design codes and algorithms for DNA composites. From an information theory point of view, it's amazing. Like the problems that we have there are fascinating, both in theory and practice. And currently what we have today is that we're doing joint work both in theory and also in practice. And a big part of it is on DNA composites.
I do have loads of questions around DNA composite, but yeah, no, absolutely. And how do you see the current trends or, approaches around DNA data storage emerging even from around the world? And how do you compare different projects? Are they all working towards the same end goal? Or do you see that all of these research projects are going to come together and we'd be seeing a unified sort of approach towards scaling DNA data storage systems?
So maybe I'll start and then I'll see what Zohar would like to add.
Go ahead.
It's a good question because I'm being asked this question many times because I say I work on DNA storage and they ask, okay, let's forget about the theory. When are you going to start making money out of it? Or, what's going to happen? When are we going to have a company? Or when are we able to buy a DNA drive? It's a good question. So basically my simple answer for this is, I don't know. I don't know how to predict. What I can say is today that we have several startup companies, as well as some of their, like Twist is also doing it. So there are several startup companies. So people put a big amount of their time and also money. So this startup also raised a significant amount of money. And just to name a few, we have DNA Catalog and Iridia, HelixWorks, and DNA-Script, DNA Algo. So there's Cache DNA. There are several DNA storage startups. And basically all of them, what they're doing is the simple application, storage, archival storage, the very, very cold storage. I even call it write once, read never, because most of the data today in the world, we barely access it. We just want to make sure that it is accessible, but we never really access it. So the idea is just to put it somewhere to know that it exists and let's not worry about it too much. And I think that's a great application for DNA storage. And this is what most of the companies or most of the community is aiming for. And that's also the way I look at it. It's like, first, let's have some device that can store data reliably. Other features like random access or something which works like really fast or I wouldn't even say like a real-time system. I see it as a secondary target after we accomplish this. There is also the idea of DNA compute. And I think it's also a good application and good motivation to work on problems. But the way I see it, let's first have some storage system that works this way. And I think some of the companies like, first of all, Twist and the DNA Catalog, Iridia, which we try to, and also HelixWorks clearly, which we try to touch base and to see what the trends and the progress is, are doing a great job. My prediction, it's a matter of like several, I would say 5 years until we're going to have, not that I'm going to buy it as a consumer, a DNA drive, but to put in a data center based storage system, which is based on DNA, to replace tapes and hard disk drives, I would say 5 to 7 years at most, if not earlier than that. And I'll also mention that the great work that has been done by the DNA Storage Alliance. So this is something that started with the Twist, Illumina, WD, and Microsoft. And I think Twist is one of the pioneers of this line. And really, I think it's a good way to see how different companies and also research groups unite together towards one target, to put some standards and to help each other to really make a big impact and to make DNA storage, not just in theory, but to really make it into practice.
Absolutely. So, Zohar, do you see DNA data storage being picked up in Israel as well?
Oh, I don't think it's a local issue. I think it will be picked up. As Eitan was saying, tribal applications are certainly going to happen worldwide. In Israel, since Israel is a leader in technology, it's going to happen in Israel as well, I'm sure. But I want to touch upon, so archival is one thing, and I think we are also going to see more sort of esoteric applications wherein DNA is really the right tools for doing this. So for archival purposes, the big advantage of DNA is, of course, the physical density and the robustness and the fact that it will never go obsolete in terms of we will always know how to read DNA because it's in our blood, literally. Okay? It's our health is DNA. So because of my background in computational molecular biology and molecular medicine in general, I know that. I know that we will always know how to read DNA. We will always improve our ability to doing so, to read and interpret DNA, to synthesize DNA, etc. So in that sense, it's very robust and therefore it will work for archiving purposes. But other than this, there's also, other applications like, people are talking about putting DNA into products as a way by which to label who the manufacturer is, maybe put, the manual for that product or for that device, instead of the manual being in paper or the manual even residing on the internet, to which you may not have access in some situations. It will be on the device itself because it's going to be encoded in DNA, which you can incorporate into all sorts of materials. I'll say a word about that maybe later. So this is another direction which I think will take off, is how can you incorporate DNA into different materials and how can you make it part of larger devices or larger systems as a component which stores information there. People are talking about using DNA for the authentication purposes. How can I authenticate that a certain, shipment of salmon fish is coming from this farm and not from another farm? Or what were the conditions under which these fish were raised, et cetera, et cetera. And this can be applied to, of course, other food products, not necessarily fish. So authentication, labeling, on product, information storage, I think these are directions that are sort of classical, not classical, but really a good fit for DNA as a medium.
And would the process be, now we'll come to the end-to-end process of DNA data storage as well, but would the process look the same when we're talking about putting DNA or, incorporating DNA as part of the product for authentication purposes? Does it need that comprehensive technology behind it as it would need for DNA data archival purposes?
Well, it will need other aspects. So for authentication, for example, we will need to develop protocols that support it. For labeling, we will need to develop technology that supports incorporating DNA into material in a robust manner. So it will not degrade over time, so it will not degrade in high temperature, or things of that sort. So this is different technology that made direction. As for coding, yes, we're going to need robust error correction codes, because despite of the fact we're going to try to protect the DNA in this other material, it will degrade. Some of it will degrade, and therefore we will have to reconstruct from a degraded version of the message we had meant to send. Therefore, I think coding theory will still play a role in that, for sure. As for physical density, also maybe not to the same extent, because we don't need to reduce that by 8, 10 orders of magnitude to be more efficient energy-wise, etc. We do want density as well. But I agree with you, it's not going to be the same parameters that we want to optimize. There will be a different set of things that we're going to look at. So in this, I just want to mention that Eitan and I are part of a consortium in Europe that just received funding for doing this type of research in collaboration with leading groups in Switzerland, in Germany, and in the Netherlands, for exactly developing different kinds of applications for using DNA as a media to store data, including these applications that use the incorporation of DNA into different materials and the use of this combined DNA material configuration in product information storage.
And I'd love to congratulate both of you all for being selected as one of the top 44 research projects. I think there were about 786 projects that had applied for this call. So it's the EIC Pathfinder call. So congratulations on that. Just talking a bit about the synthesis and sequencing technology, and both of you all play quite a pivotal role, and you've published the information as well over the period of time. Zohar, you've been involved in many DNA synthesis technologies over the past several years, and you've probably been quite pivotal in those development of those technologies as well. What are some of the current DNA synthesis technologies, and is there a way to see if a certain method works better, specifically for the application of DNA data storage?
Okay, so first, in terms of what technologies are emerging and what we can expect, I think there may be others that, there's activity going on that I'm not necessarily aware of, but certainly there's a lot of activity around enzymatic synthesis, which would be more cost efficient than the chemical synthesis approaches that are mostly based on the brilliant phosphoramidite technology, which is what made synthetic biology in general possible. Not only storage in DNA, but all this field of synthetic biology has really gained a huge boost from Marv Carruthers' work on phosphoramidites. But again, enzymatic synthesis will probably TDT and other enzymatic approaches may be things that we're going to see emerging as methods for generating synthetic DNA, again, not only for storage, but also for other purposes. I think the direct synthesis of longer mers, what do I mean? Not so phosphoramidites that are not necessarily single bases. Maybe we can go in the other direction. So can we synthesize base pairs? So people are working on directly synthesizing base pairs, but also directly synthesizing dinucleotide, trinucleotides, 5 nucleotides, whatever, longer mers that can have really interesting implications in storage. We can touch upon including different types of composite DNA layers. So that's another direction. And I think the most efficient methods currently are based on what I will call the Agilent philosophy of using basically a printing technology. So the synthesis is done as a printing process. This is also what Twist is doing. It's not the same as Agilent. Twist has really made great progress in terms of how efficient the process is, et cetera, and really focused on that. But it's the same philosophy as the printing philosophy. But, I think we're going to see photolithography maybe coming back and more efficient approaches to photolithography, not the Affymetrix ways. So I would call it photomediated synthesis, not necessarily photolithography. So no masks used. So it will be approaches where you can direct the light to the points you want the synthesis to happen on in a programmable way. If you have to generate masks, etc., again, on Affymetrix, that this will be very expensive and inefficient, and it's not customizable, and I think this is why this approach failed. But other photo-enabled approaches, I think we're going to see them coming back. Maybe other flooding synthesis technologies that will not require. So the printing approach, basically you have to go to every oligo and do something at that oligo in its own time, more or less. You can parallelize a little bit. But I think the next generation of synthesis technologies will have greater parallelization rates. So you're going to do something over a large group of oligos, and do something else over a large group of oligos, et cetera, and everything will be much more efficient.
Eitan, what is the effect that you see then on when you talk about the encoding and decoding sort of methodologies? What impact does synthesis have on the DNA data storage system?
Right.
Just
so
in
that's
the
a great question. Maybe Eitan will expand on this, but because this is exactly the point that Eitan made before, that by understanding the error mechanism of the synthesis, we can understand what kind of error correcting means, codes or other means, we should be using in order to make this a useful technology. Okay. So as Eitan mentioned, one of our first work together basically was to try to develop. So we developed software that enables you to characterize the error mechanisms in a certain synthesis run. So I think we will have to understand that. And of course, different synthesis approaches will have different error mechanisms. And by understanding the error mechanisms and tailoring error correcting codes and other error avoiding approaches to these synthesis methods, we're going to be able to make the entire pipeline more useful. But maybe Eitan wants to say more about that.
I can only agree with everything that Zohar said. A little bit more, I don't understand all the different type of synthesis methods. The only thing which I can say, and this is also what happened in the past in hard disk drive and flash memories. Flashes started with transistors of the size of like 400 or 500 nanometers. Today, the size of the transistor is below 20. But by shrinking from 500 to 20, it's not going to work as well. It is much more noisy. So it's a very simple story. Like you, in the same area, in terms of capacity, you can load and you can store more bits, but the bits are more noisy. But it's a trade-off. As long as you, in terms of information bits, you can still store more information bits, it's a winner. So that's exactly what has been done in flash memories and hard disk drive. They're able to put more bits in the same area, but the number of errors increased, or the error probability increased. Then using coding scheme, you can still compensate, and still by the end of the day, it is possible to store more data. And this is the end of the story. This is the summary of the story, how it works. Now, when the industry shifted from hard disk drive to flash memories, the transition was not too complicated from the coding point of view, because the same solution that they use for hard disk drive in terms of error correcting codes pretty much also works for flash memories. DNA storage is a completely different story. We can basically take nothing that has been used for hard disk drive and flash memories and migrate it as is to work on DNA storage. And currently with some of the existing experiments today, this is what people have used, like a Reed-Solomon code, LDPC code, but simply because we don't really have other solutions. It's embarrassing to say as an information theorist and coding theorist, when Claude Shannon had his first paper, like the 1948 Fundamental Limits of Information Theory, he basically set the foundation of what is and is not possible in terms of error correction to correct the noise. And it was very simple and elegant. And he mostly talked about what's called the binary symmetric channel. Once you transmit bit, every bit could be flipped from 0 to 1 and vice versa with some fixed probability. To understand what is the limits of this channel, he already solved it. It was very simple, basically understanding the capacity of the binary symmetric channel. Yet to build codes which achieve this channel, that was not easy. And only Polar codes are the first codes that exactly, or maybe LDPC in some level, but actually to reach the capacity of the binary symmetric channel is fully solved. And today, if we want to design codes that can correct what we call substitution errors, it's a done deal. We know how to do it. Basically, every length, number of errors, worst case, not worst case, fully solved. In DNA, we also have the insertions and deletions. Now, if I want to design a code today that can correct one deletion, the problem has been solved 50 years ago. If I want to design a code that can correct two deletions, meaning if I'm going to transmit 100 bits and 2 bits are going to be deleted, meaning I'm going to get 98 bits, and I want to design a good code for that, I don't know how to do it. It's as simple as it is. Maybe I can still do something, but if somebody asks me, what is the best code that I could use today? I don't have an answer for this. And this is why when we do experiments today, we don't have a good coding scheme to correct not only deletions, we're talking about deletion, insertion, and substitutions. And it's one of the, I would say, highest priority that we have today from the coding and information theory communities to design code specifically for this target. We don't even know what is the capacity of this channel. It's an open problem. We don't even know how to do it. There are so many open forums only for this. And in the DNA storage, it's not only this, because we also we work with short trend and we don't have order for them. And we still have many copies of them. So this is only the first layer to design code for insertion, deletion, and substitution. And this is only the beginning of the work that we have to do here.
So is there a way to sort of evaluate efficiency of a system, Zohar, it probably goes back to your work as well, and it's probably where the composite DNA letters also sit in. Do you want to tell us a bit about it?
Yes, so I think, there are, again, from a practical point of view, the most important performance indicator, if you like, for any system would be the cost. I mean, we would like to achieve as many bits of storage in a certain level of robustness, etc., at as low a cost as possible. So, and cost is affected by many parameters as well. I didn't mention before, I'm sorry, I will go back to synthesis for a minute. What I didn't mention, another direction that is happening is the orthogonal system. So maybe something which is not DNA, which will be like DNA. It will have the hybridization properties, it will have the complementarity properties, it can be synthesized, etc., etc. But it so happens that this is not the same molecule as we have in our blood. So yes, that's interesting. Maybe if it's far, far more efficient, it can be interesting. But I think we have to always remember the great advantage of DNA, which will always be something that we know how to handle. And I think that, so I emphasized this point before, I will emphasize it again. I think this direction of research of orthogonal systems can make the systems much more efficient. So it has a potential, but we have to remember this caveat about it. So again, this goes back to what are the parameters that we should be considering. So cost will be one thing which is influenced by the efficiency. And then there's robustness. So robustness has several angles to it. One is, yes, will it survive, high temperature? Will it survive 1,000 years? Will it survive being crushed in some physical way? Will it survive different conditions? That's robustness on one hand. And the other aspect for robustness is, will it survive? Will there be technology that will allow us to read it? Okay. And also, even if it is degraded, can we read it despite the fact it's degraded, because we have good error correcting codes to offset that degradation. So, and I think to go to the point of the composite DNA letters, what we address in the composite DNA letters, and this is something that is being further developed, is to basically increase the size of the alphabet so that in every synthesis cycle, I get more bits that I can store in that cycle. And we want to do that without paying in the other parameters. So one place where we are certainly paying in composite DNA is the number of molecules that we have to basically generate in order to support the code. So because the code uses, so maybe I will just say what composite DNA means. Composite DNA means that in a certain position, I don't just have the alphabet A, C, G, and T, but I'm going to add letters to this alphabet by also considering combinations. So, for example, if I consider all combinations of 2 letters, then I basically have a 10-letter alphabet. Okay, what is a combination? Well, a combination will be represented by the fact that at a certain position, I see 50% A and 50% T, say, or 50% C. So if I see 50% A and 50% C, and of course 50% doesn't have to be 50%, it can also be 47 and 53. So if I see this combination there, that means it's a different letter. It's not one of the 4 basic letters. That's the basic idea, okay? And now it can be projected to doing much more complicated things, which are things that are being pursued, for example, by other composite codes that we are developing currently with Eitan and other students in both my group and Eitan's group, as well as by Helix Works, as well as by Catalog. So there are different efforts in the world to further leverage this idea of composite DNA. But with that comes the price that you have to have more molecules, because I cannot infer 4753 if I don't have at least 100 molecules, for example. Whereas if I just needed to say A and I saw 10 times A and 1 error in the sequencing, then I know it's A. Okay, so I will need more molecules. So where is the trade-off between the slightly increased physical density and the much improved, what we call logical density, so number of bits per synthesis cycle. So this is just an example of the kinds of trade-offs that we need to take into account. Of course, there will be the coding questions. So if we want to put this into a tree or into a salmon fish, should we use this because the coding will be more difficult at the end, the decoding will be more difficult at the end, or will it not be more difficult at the end, etc.
100%. And you've also developed a novel method to encode information, which is again using the combinatorial encoding and short data synthesis. How does this approach stand against other methods? And do you see this replacing other methods, or is it, how you said, it depends on what the end result would be, or what is it meant to be used for?
Yeah, I think that eventually, I think, so there's a trade-off, okay? Once we start being more sophisticated with the encoding, we have to also be more sophisticated with decoding, and we have to also have an agreement of what the encoding and decoding had been. Okay, so it's not ACGT anymore. Even with ACGT, I have to have an agreement that A is 0, 0 and C is 0, 1 or whatever, right? I have to have some, because someone may do it in a different way. So even with the binary code, the ASCII code, right, which translates binary code to letters in the English alphabet, we have to have an agreement, but this agreement is sort of standardized in the world now. But all I'm saying is that, yes, we will always be able to read DNA, but how do I interpret that string of DNA as Beethoven, as some music by Beethoven? That's an agreement we're going to have to reach.
Yeah.
And there will have to be an agreement which is broadly supported, and that's why there are efforts in the world now already led by storage people as well as DNA storage companies that we will have to sort of develop standards in this respect. And there may be different standards for different applications.
Yep.
The more standards, the more the confusion, the less standard. On the other hand, the more standards, it's more fit for the application. So I think it's early days. I don't know yet what's going to happen with that.
It's yet to be developed. Eitan, I have questions now around both like the encoding side and decoding side of things as well. So the way we store data onto DNA works quite differently, even from the point of synthesis and sequencing. They introduce a lot of errors, as you've mentioned before as well. How would you first explain it against other storage mediums when you talk about DNA data storage just as a medium? How does it stand against all other mediums like flash drives and hard disks and all?
So the first fundamental difference is the type of errors. So in hard disk and flash memories, as we said, mostly or only what we see is substitutions. If you store 2 kilobytes of data, you're going to read 2 kilobytes of data. Some bits are flipped, but it is still 2 kilobytes of data. You store, you read it, you put some error correcting code. The framework or the story is relatively simple. You just need to design a code for this. When we do it in DNA, nothing is the same, basically. So first, the type of errors are insertion, deletion, substitutions. That's one difference. We cannot generate, at least as of today, long oligos. So the oligos have relatively short length, up to, say, 300 bases. So that's significantly smaller. So if we have a big file, we have to store the file in many. The many could be like hundreds of thousands or even millions of these oligos. And each one of the oligos is going to have many copies. And all of these copies are basically stored as a noodle soup. So everything is tied inside this container, completely out of order. And then we want to read, so we take a sample and we read a portion of everything that has been synthesized. No other storage system or memory system today works even barely close to what we have here. So first, insertion, deletion, substitution. Second, lack of ordering. Third, we're talking here about shorter vectors that we can store. And now we have to design codes and algorithms that basically have to work with everything together. And we also need to take into account that everything is technology dependent. If you have a different synthesis method, the errors would behave differently. If you have a different sequencing method, the errors behave completely differently. So today you can sequence with either Illumina, like the mainstream, I would say, Illumina, or nanopore sequencing. So nanopore sequencing, this is what we have in my lab. So we wanted to make sure that we can sequence by ourselves. And so Omer Sabari, my PhD student, built a lab that he can work with nanopore. So it's really helpful. It's really great today that we can work with the raw data, so we can sequence by ourselves. And now it's another different story because it's not that you just read the bases. We have some soft output. We get some signal out of this nanopore, and the signal should be converted to this ACGT, but what's called the base caller. So now we have another dimension, how to build a really good base caller, specifically to how we read and we store data today in DNA storage. Most of the base callers today are more designed for other type of DNA, whether it comes from nature, for a human being. And it's a different type of data that is stored today in DNA. Maybe we can store something which is going to be easier to analyze and to process once we read it with the nanopore sequencing. So and that brings another dimension. So the type of problems and the challenges that we have are fascinating. They are intriguing, that's for sure. That means that we have a lot of work that has to be done that comes to my group, Zohar group, like the concern that we have together. But basically, all of this community is trying to push DNA storage forward. Like many things have to be solved. This is just the beginning of this road.
Absolutely. And just in terms of, like you mentioned, retrieving data then when you have so many errors can be quite tricky as well. How do you think we can prevent, or like how can we prevent lesser errors during these steps? Is it just via the correct coding methods, or how do we go about it?
I think it's going to be the opposite. We're going to have more errors. And it's the perfect example of Zohar was talking about the composite DNA. You have a way to increase the density so we can store more data. And you don't need anything special. It's the same synthesis method, but this is a really nice, elegant, and a smart way to increase the information density. But it's a trade-off, because once we do it this way, we can reduce the cost. This is wonderful, but the type of errors is different, and we're going to see more errors. So what we're going to see probably in the future is to see this kind of method and maybe some other ones. We have to think outside of the box and to design some other schemes that will help us to improve the density by this, meaning to reduce the cost, which is the main bottleneck today for the cost of synthesis. And once we do that, the type and the number of errors, in my opinion, are only going to increase. And then we have again to think outside of the box to design the good coding schemes and algorithms to compensate and to design the good codes to correct these errors.
I think I want to add something to this. So you mentioned before the short-mer synthesis that we're developing now. So that's an example of where it's a system that can take advantage of the basically phosphoramidite synthesis philosophy. Okay, so it's efficient synthesis, but it uses not a single base composite DNA, but something that is based, say, on trimers or on dimers or even longer. Not too long. That's the idea. Don't make them too long, because then you lose this advantage of efficient synthesis. But once you do this, we introduce other types of errors, as Eitan was mentioning, and I will need to see even more molecules of each one in order to understand which molecule I had seen. The advantage is if we understand the error mechanism, which we intend to do, so if we understand what specific errors happen more than other errors, we can also design more efficient codes to deal with it.
So what are some of the already existing codes to correct these errors? And Eitan, this is probably your work as well around the deep neural networks. Do you want to talk about it?
Yeah, when you said these errors, as you can see already, so just by the last
Just
example, so make my life even more difficult. As if a DNA composite is not challenging enough, now you have to do it by shorteners. So it's
That's part of my part of the collaboration.
Zohar is gonna equally contribute to make your work
yeah.
easier.
And this is why like it's important to work with people who understand and people who know how to make it like much more efficient and to reach out. And so this is why this collaboration is so successful, because it's we really complement each other, and that's how I see like as a really fruitful collaboration, not just to work with somebody from your field, really to work with somebody outside of the field and to find like the mutual interest and topic to collaborate on. But going back to your questions, so what kind of coding schemes should we use? And again, there are some ideas like to correct insertion, deletion, substitutions. We already have some schemes like watermark coding, and some of these LDPC type codes can also correct these synchronization errors. Polar codes have been proposed by Erdal Arikan in 2009 as capacity achieving codes for the binary symmetric channel. That being said, they're also being used nowadays to correct deletions, insertions, and substitutions. So we are not completely clueless. We do have some initial ideas where to start from. But you see, we have to keep up with the technology, and we have to know where it's headed, where it's going from. And every time when there is going to be a new advancement or a new idea, we have to adapt the solution and to see how to first to understand how the errors behave, and to design not only the error correction codes for this, because everything comes together with the entire system. So the entire system, because we have many strands that we have to synthesize. So the first task, I call it a clustering or binning, that you have to partition the group, all the reads that you have into groups that they all came from the same oligo that we wanted to synthesize. And if we do it together with the composite, it is more challenging. Then the next step is what we call reconstruction. So if even let's put composite aside, if I'm going to have many copies of the same information strand and they're all noisy copies, so how do we take this majority, or how do we make the conclusion of what is the correct information strand once we have several noisy copies? It is still basically an open problem how to do it efficiently. So one of the nice schemes that we did recently in my group is we use the tools from deep learning. And because to specifically correct errors for this, it is not necessarily easy. And we also find deep learning to be useful for this nice problem. And the idea is to train a system that can understand how the errors behave once we synthesize in one specific method, or to build a simulator that can simulate how the errors behave, and then to make the system train and to understand how the errors behave, which is challenging by itself, because how do you make a simulator that actually simulates how the errors behave? It's a challenging task by itself. But then the idea is to build a network that can understand and learn how these errors behave. And by this, what we wanted to do is to build an algorithm that will work, first of all, efficiently in terms of speed, but no less important, also accurately. And that's something that we are still working in my group. And we hope to submit a paper very soon that uses these deep learning tools in order to be able to correct errors in DNA. This is without composite. Really, the next step is to do it together with a composite on the base level and also in the shortener levels.
So will it be recognizing patterns and schemes as to what these errors are introducing in the data storage system?
Yes. And again, we're coming back to the beginning. Once we understand how the errors behave, it makes our life and work easier. If you don't have a deep understanding how the errors behave, how can we design something how can we correct something that we don't even know how it behaves?
So it's all about understanding first. It's basically laying the foundational work to kind of build on then.
Exactly.
So actually that brings me to the next set of questions, which are probably very interesting to both of you. It's about understanding the challenges that are associated with DNA data storage and how are they being addressed? I don't know, Zohar, if you want to go first and, talk about some of the challenges that you're immediately facing. And actually, even before that, what are some of the areas where both your research groups are absolutely interested in when we speak about DNA data storage? Would it be end-to-end, or are there parts within DNA data storage systems that your groups are working on?
Yeah, we don't have the capacity to do, commercial scale or production scale end to end. We have the capacity to maybe develop a proof of concept solution end to end. That, yes. Even that, we may need the help of others, for example, for the synthesis step, where we don't do the synthesis ourselves. So we're working with collaborators who do that, either in industry or academia. So, and we can design the proof of concept. The proof of concept will have many aspects. It will have the coding and the design. It will have maybe the robustness of the storage. And then it will have the error correcting code and the decoding at the other end. So I think we're interested. I think in this sense, I'm speaking for both groups. So we're interested in all aspects of this process that has computational questions, information theory, probability theory, data science questions involved. Everything we can use, coding theory, information theory, etc., to make steps in this process more efficient. We're interested in that, and we're investing efforts in improving these aspects. That's what we're doing. So this goes from design to coding, and design and coding, as I said, will be, or as we both emphasize, will be dependent on the actual synthesis and the error mechanisms, on the actual storage and the error mechanisms. Maybe something, when you put into a tree, maybe the degradation mechanism is different than when you put it into a container and then just store it somewhere. So I would say, and in terms of the challenges, again, it's the same thing. I think that the main challenge, or the main current limitation, and maybe a challenge for storage on DNA, is what I would call the immediacy. There's no immediacy. And I think it's clear that, at least in the short term, we cannot expect storage on DNA to work like our cell phone works. Okay, we don't. If you want to see, a document that we had saved yesterday, and we want to see it today and again tomorrow and change it, and that's probably not the right application. The applications will be in archiving, as Eitan was saying. Write once, never is maybe strong, but sort of never read. Hopefully never read, but if you need to read, you can read it. And then in these other more vertical applications, if you like, of labeling, of applications, or protocols based on that. And there the challenges are, there are challenges in all aspects. The cost of the synthesis is a challenge.
I was going to come to that, because every time I have this discussion with any of the researchers, and when I ask them, what do you think would be the immediate limitations, especially when we talk about scaling DNA data storage from lab to market, it's always about the DNA synthesis. And it's not so much about the sequencing aspect. And I'd like to know why not. Why don't we talk so much about sequencing? And I'd love to hear both of your views on it as well. I know we are, like, when you talk about long-term archival, we're speaking about something that we may or may not need to read. But there could be instances where, sequencing might be a part, like decoding the information might be necessary as well. So how would you sort of explain that limitation?
Anyone go first?
Sure. I think sequencing has received much more progress, like in the past 20, 30 years than the synthesis. So that's sort of, and mostly because there was much more need for sequencing than the synthesis. And as of today, this is still the case. And though I feel like the DNA, it's not like we have to use DNA for storage. It's a really nice application. But whether this is the most important thing that we have to solve, No, I mean, we can still survive, I think, without the DNA. I think it's gonna make our life much more green and better if we can store data inside DNA. But it's not like as crucial or mandatory as it's like for sequencing, as we had like the genome project. So, and I think for sequencing, we already know at this point it's mature enough. We know what can be done or what even cannot be done. And there's still a bottleneck in terms of limitation of how fast we can read DNA, how much this is going to be improved, and whether we can have it as a read-time system. That could be one of the limitations. And but also we can still live with this limitation. We can still build everything around this because it's again, if it doesn't work on your cell phone and it's not a real-time system, it is still okay. There are still many other applications.
Could you
And I
give
think
us
that
could you give us a time of like what is the read or write speed at the moment?
for to read DNA,
Yeah.
megabyte per hour, if we're lucky.
And
No, I
It's
think it's a setup time. You need to,
Yeah.
you need to have sample preparation. It's hours, basically. It's
Yes.
hours. If you want to open a file, it's hours for sequencing.
But it's also because of the preparation time.
Yeah.
If you think, but let's just, if I put the preparation time aside, let's assume it, but just by putting this inside the nanopore and
Yeah.
still making the base corner and everything works, it's on a matter of seconds,
Correct. No, not
Yeah.
days, but yes,
minutes.
maybe 1 hour, or
Yeah.
maybe if it's a short file, if you manage to, pick up by PCR or something like this, it's what's called random access machine. If you manage to do a RAM trick and you manage to take only the file you're interested in, maybe you can do it in 30 minutes. But I don't see how it can be done in much less than that. So even if everything is optimized, and that's the sequencing part, you are right, that's the sequencing part. And then there's the synthesis part. But in any event, that's no competition to, opening a Word document, which will take under a second. Right? The applications will be according to that. There are many things where DNA can have an advantage. So on the other hand, if there's fire, the Word document is gone, while the DNA is not gone.
No, absolutely. But before we leave here today, I think that's a lot of nuggets of information. But I think one of the things I'd love to learn is that we see an increased amount of partnerships and collaborations happening between academia and the industry as well. What roles do academic institutions play when we talk about bringing such emerging, innovative, novel technologies from lab to market? And how do you see this evolving? How do you describe that role?
Let's start, if that's okay.
Yeah, go ahead.
So I think one thing that we saw, at least I saw, when people from academia started to work on flash memories, is that it was too late. It was too late in the sense that the technology was already mature enough. So if we wanted to influence or to bring some impact into the market, it was already too late because the technology was mature and already fixed. That it was very, very hard to make any changes in product. Now we do have, this is the baby step. We do have the opportunity, we do have the knowledge, and we should be able to contribute and to make sure that we take the knowledge and to bring it into the product. Also because we can see it is necessary. It is necessary to work together both from academia and industry. And we have to make sure that there is a knowledge transfer of whatever we know in academia and to bring it into the product of DNA. And it has to work this way. It is very challenging. It is not easy. There are many, many problems that have to be solved here. And just working solely industry or solely in academia, we're not going to make any impact this way.
How do you feel
I
about
think academia basically can do, as I said before, the proof of concept, and then if it's interesting, industry might pick it up and make it broader and bring it to a broader usage. I'd like to look at synthesis or microarrays, for example, where they started in the academia or where they started in the industry. So, I was at Agilent in the early days of microarrays. Did Affymetrix and Agilent develop the first generations of microarrays, or was this Pat Brown at Stanford who basically did the first yeast microarray, which was based on cDNA, by the way? It was not based on synthetic DNA in a sense, or not synthetic DNA. So it synthesized based on naturally occurring DNA. So it's a question. So I think Pat Brown and David Botsin at the time had the advantage of doing the first application proofs of concept. And in parallel to that, Agilent and Affymetrix and other companies optimized the synthesis technology to make applications broader. And then when other applications came, which are not expression profiling, such as copy number measurements, for example, it was already industry that led the actual production, but still academia that led the proving that the application is good for something. So can we use it in the clinic? We want to show that there's, health medical implications of what the measurement enables. I think it's the same kind of interaction that will happen here as well.
Do you see there's been an improvement in between the communication between the academia or the industry? Is it much more collaborative than before?
From my experience, at least, I think it is. If I compare it to other memories and storage industries and communities, I would say yes. In general, the more mature the technology is, the less interaction we have between industry and academia. Many times because industry will not share the secrets for obvious reasons. Will not share their own secrets with academia. So then it's hard to make impact once you don't know, as we said all the time, how the errors behave or how the system is structured. So now with DNA, everything is open. So everybody can work on it. And I wish it were the case also for other memories technologies, but clearly it doesn't work this way, and that's understood. In general, I would say that the memories in storage industries are tough. It's very hard to compete. Even there are many great solutions that came up in the past for other type of memories and storage solution, like holographic storage was a great idea back in the late 90s and the beginning of the 2000s, and eventually did not make it into the market. Like there was still a product. I want to believe that DNA is not going to be a similar case. I feel like people do recognize the potential that we can have in DNA. And there is a lot, as we can see, a lot of effort both from industry and academia. Let's see what's going to happen in 5 years. I want to jump 5 years forward to see what's going to happen.
Yeah, I'm just going to ask you do you think DNA data storage might become mainstream, or, that would be probably the thing to see, just at the minute, I don't think we are able to visualize it beyond the schematic diagrams. It would be really nice to hold, would it be a drive? Would it be just a coating? Like Zohar mentioned, is it going to be a physical ID on top of all the products that we consume? It's really an interesting area to be involved in at this point. But do you have any announcements or news to share, something that you're excited about that's happening in your lab before we leave here today?
Yeah, I think we shared the news about the project. We're working on lots of exciting things. I think we mentioned these directions. We're going to see news as we go. Yeah, I don't think there's any announcement that I'd like to make on my side at this point.
I think it's as I usually tell my students, the best is yet to come.
Yeah.
So, and I feel it's really this case. I mean, it was really good news to have this consortium of Dydex with the other collaborators. But I'm also, when I talked to Zohar before that, what I was mostly excited about this is the ability to collaborate with the other group members that we have, because this is no less important than just receiving this funds that we have which is highly appreciated by the European Union. And the fact that DNA storage was one of the topics for the Pathfinder this year just tells everything. But no less important, or I would say even more important for us, is to make sure that we can work with, for example, with Robert Gratz from ETH Zurich and Mark Somoza from Munich, and to really make an impact. It's not enough just to work on your own. We have to make sure that we can collaborate and also working with industry, for example, like Helix work or Twist. Only together we can make a true impact here.
Thank you so much, Eitan. So thank you so much, Zohar, as well. Thank you for sharing your insights and shedding light on the potential of DNA as a storage medium and the exciting possibilities it presents for the future of data storage. Thank you for listening to this episode of Dive into Deeptech Moss, powered by Helixworks Technologies and brought to you from Republic of Work Studio, Cork's leading innovation hub. Follow us for more updates. Until then, stay tuned.